Well, officer, I can remember it crystal clear. I was enjoying a delicious juicy steak in Mario's on 24th Street. Nice and rare, just the way I like it. With a little spinach salad on the side. When out of nowhere, he attacked me.
Miss Portelli, can you describe his general morphology?
He was a tiny, broad-shaped pink fellow, about 2 micrometers tall and about 1 micrometer wide. One of those gram-negative bacteria. I remember, because I tried to zap him with my antibiotic taser, and it didn't faze him one bit. He was covered in fimbre and had a long flagellum he was whipping around. I think he was a Ute, a young E. coli, maybe about 50 years old or so. He was a non-sporulating prokaryote. If I saw him, I'd recognize him in a New York second.
All right, Miss Portelli, I think we might have your man. Send in E. coli strain 0104H4. Step forward for identity.
Bad boys, what you gonna do? What you gonna do when they come for you? Bad boys, what you gonna do?
That's him! That's the joint that gave me explosive diarrhea and almost killed me. Let me at him!
Easy, Miss Portelli. We'll take it from here.
Bad boys, what you gonna do? What you gonna do when they come?
Escherichia coli, aka E. coli, have 2 pathways to commence a life cycle: binary fission or conjugation. The steps of binary fission are as follows. In bacterium, before binary fission, DNA is tightly coiled. The DNA of the bacterium then replicates. The DNA is then pulled to the separate poles of the bacterium as it increases in size to prepare for splitting. The growth of a new cell wall begins to separate the bacterium. The new cell wall fully develops, resulting in the complete split of the bacterium. Finally, the new daughter cells have tightly coiled DNA, ribosomes, and plasmids. The steps of conjugation are as follows. When 2 bacterial cells are in very close proximity, a donor cell produces a pilus.
Oh my!
The pilus attaches to the recipient cell and brings the 2 cells together. The mobile plasmid is nicked, and a single strand of DNA is then transferred to the recipient cell. Both cells synthesize a complementary strand to produce a double-stranded circular plasmid and also reproduce pili. Both cells are now viable donors. You're a real F+, baby!
You're not so bad yourself, my F+ sugar daddy.
E. coli have the potential to live for hundreds of years.
So what happened? Where'd they discover you, Joey?
Well, it all started back in 1885 when this German-Austrian pediatrician by the name of Dr. Theodor Eschrich believed he could figure out the ailments suffered by kids from studying bacteriology. He discovered this while researching the cause of fatal intestinal disease in children. That guy actually examined baby doo-doo, looking for responsible organism, and he found E. coli, quickly growing and multiplying. They named us after him, after the poor sap kicked the bucket.
No, you fimbre head. Where'd they find you?
Oh, right. Well, once the old lady ingested me, I used my special protein to help me survive. At the end of each of our fimbre are special proteins called adhesions. The specific type of adhesion varies by type of bacteria. But regardless of the type, adhesion molecules allow bacteria with fimbre to adhere to host cells by docking, like a lock and key, with the receptor proteins on the surface of the host epithelial cells. This ability of fimbre to stick to epithelial cells leads to many diseases transmitted via mucous membranes. This tactic allowed me about 7 days to hide out in her system, till she finally flushed me out. How about you? You being a good bacteria? Where you been hanging out?
You know my ecology. I like hanging out in the gut. Peoples, animals, any warm-blooded GI tract where I can help out. You know my cousin Vinny? They found him and his family colonizing an infant's GI tract within hours after it was born. Much better than the more commonplace our kind are found in the human colon. You know, we're the predominant facultative anaerobe there. Joey, I heard you was hanging out in some dirty produce again. I wish you'd be a good strain and settle down with me. We could share a nice symbiosis somewhere.
Come on. I can't give you that, Lola. I'm not a good strain. I just can't see myself in symbiosis, like those good strains, hanging out in the GI tract, in a safe, enclosed, and comfortable living environment where bacteria receive the required nutrients for reproduction and growth and carry out several necessary functions. A place where good strains, in turn, make it possible for humans to absorb vital nutrients, including vitamin K, through the colon. You want me to be an essential organism in the human body? I can't do that. I'm a bad strain. I'm 0104H4, toots. I'm in the business of making people sick, not helping them out like them was other good E. coli. Go find yourself a good strain. Go on, get out of here.
Oh, Joey, how could you do this to me? Ooh.
Thanks for coming to dinner with me, Lola. I know you love glucose, so I thought the stomach would be the perfect place, and the bile here gives your cell membrane a beautiful glow.
Oh, no, but you're so romantic.
The metabolism of E. coli is quite diverse, with several options. Being heterotrophic, their main source of carbon comes from glucose from the host. They may exercise central metabolism, which employs the following three steps. First, the EMP pathway converts glucose to pyruvate. Next, the TCA cycle oxidizes acetyl-CoA to CO2. Lastly, the PPP cycle oxidizes glucose to CO2. Another pathway in which E. coli can utilize the carbon from gluconate is where it metabolizes through the Ettner-Duderoff pathway, where eventually pyruvate is converted to CO2 and acetaldehyde, which is further changed to ethanol. The third option would be to convert pyruvate that it generates through the EMP pathway into ATP through fermentation, which makes sense since E. coli is a facultative anaerobic organism.
Dinner was lovely, Norbert. I had a great time.
Me too, Lola. How's about we go back to my place for a nightcap? I've got some nice maltose we can wash down with some lactose.
You read my DNA. Let's go. Norbert, he's the greatest. I can't wait for him to meet the family. Here's our phylogeny, the good old family tree. Way off, barely related, you can see the eukaryotes and the archaea. But here, in the bacteria branch, is where my closest relatives lie. My phylum is with the proteobacteria. We are a major group of negative bacteria, including a wide range of pathogens. We have our alpha proteobacteria, which grow at very low levels of nutrients with unusual morphologies, such as stalks and buds. Some of these guys even fix nitrogen. Then we have the beta proteobacteria. These guys often use nutrient substances that diffuse away from areas of anaerobic decomposition of organic matter. We even have some chemotrophs in this group. Lastly, we have my immediate family, us E. coli, come from gamma proteobacteria, the largest subgroup of them all.
Because of its manipulation and long laboratory history, E. coli contributes an important role in current biological engineering. It has been vastly used to synthesize DNA and proteins. Most results from E. coli research can be applied to humans and animals. The most useful contribution of recombinant DNA from E. coli is to use the manipulation of E. coli to produce human insulin for diabetes patients.
You're welcome.
Since E. coli can produce human enzymes through recombinant DNA techniques, it is widely used as a very good tool to produce useful compounds or enzymes for medications. In some studies, scientists are exploring new methods to cure Alzheimer's disease with the use of E. coli manipulation.